All right. Are you ready, Ali?
I am ready.
Okay, I'm going to get us started. Okay. Good morning or good afternoon, everyone, depending on where you're dialing in from. This is George Orling from HDSA, and I'm really excited to introduce our next HDSA research webinar speaker, Dr. Ali Koshnan from Caltech. But before I do, for those that may be new to the research webinar series, I just want to, or to the Zoom platform which we're using, it's super easy. If you want to ask a question, you basically just click the chat icon shown below. At any point during Dr. Koshnan's presentation, you can ask a question. We'll get it here, and only the HDSA and the panelists will be able to see your questions. So feel free to type whatever you like, and we will ask them on your behalf at the end of Dr. Koshnan's presentation. Next month, before I go on, we will record this or archive this presentation, and you'll be able to view it again. Just you have to dial off, or you'd like to send to somebody who can't participate today. We will archive it and put it in our YouTube channel, which can be found on our brand new website, hdsa.org, by clicking the YouTube icon at the top of the page, which I've highlighted here in a red oval. Next month, in June, we're not going to have our monthly research webinar because we're going to have a plethora of research presentations being live streamed during the HDSA convention in Boston, which is taking place on June 27th through the 29th. And in particular, the entire research forum on Saturday, June 29th is going to be recorded and live streamed, so anyone can participate. Here you can see a really exciting agenda that we have lined up. We're going to be hearing, which is super exciting, from Dr. Craig Mello, who is a Nobel laureate, who was the man, one of two men who won the Nobel Prize back in 2006 for identifying RNA interference, as well as Dr. Anastasia Korovora from UMass, who is working closely with Dr. Mello on a project related to Huntington's disease. Then we'll, from 11 to 12 on Saturday the 29th, you'll hear from Drs. Ed Wild and Jeff Carroll. They'll be giving their research year in review, kind of HD Buzz style. And then, as if that wasn't enough, we'll get clinical trial updates from the two Huntington lowering trials that are currently recruiting in the United States, one from Wave Life Sciences, the Precision HD1 and Precision HD2 trials. You'll hear from Dr. Serena Hung, and then we'll hear a Roche Genentech presentation from Eric Lundgren and Dr. Lauren Boak on the open label extension, natural history, and the Generation HD1 trial. So hopefully you can join us either in Boston or online for this really exciting day of research on June 29th. Excuse me. All right, so with that, it gives me great pleasure to introduce Dr. Ali Koshnan. Ali is a senior investigator at the California Institute of Technology. He received his PhD from the University of Texas Health Science Center in San Antonio. He was a Howard Hughes postdoctoral fellow at University of Southern California and an NIH senior fellow at UCLA. He joined the laboratory of the late Professor Paul Patterson in late 2000 and has worked as a senior scientist on various projects related to Huntington's disease, autism, Rett syndrome, and stem cell biology. His current research is focused on understanding the pathogenesis of Huntington's disease and Rett syndrome. Fun fact about Ali, I don't know if you know this, Ali, but Ali is the only two-time recipient of the HDSA HD Human Biology Project Fellowship that we've been running now for about 7 years, and Ali has been the recipient of 2 of those fellowships. And finally, you know, Ali's going to be talking about, the title of his presentation is Gut Microbiota and Huntington's Disease. HD has long been referred to as a brain disease, and it is, but you know, as we all know, the mutant Huntington protein is expressed in all organs in HD patients. So it makes sense that it is likely kind of wreaking havoc in places outside of the brain. And it's been a very underserved and under-researched area, and that's why we're very excited to have Dr. Ali Koshan here to present his work on better understanding what's going on in the gut and GI system of HD patients. So with that, I'm going to turn it over to you, Ali. You can take control and share your presentation.
Do you see my slide, George? Can you hear me?
Yes. Yes.
Hello?
Yes.
You can hear me?
Yes. And we see your.
Okay, great.
The floor is yours.
Wonderful. So thank you so much, George, for the introduction, and thank you to all of you for attending here. I'm really excited to be here, and as George mentioned, we are going to discuss gut microbiota in Huntington disease. A pretty tall order, but I think it's a really, really exciting field and with a lot of promises for the therapy towards the Huntington disease. So to be on board, usually, microbiota refers to a collection of microorganisms that they reside in a system. For example, gut microbiota refers to this collection of critters that they live and reproduce in our guts. So for the entire talk, I'm going to mainly focus on gut microbiota, but knowing that there are many organisms and other body sites that also are as important. To note that animals cannot survive at least long without a microbiota. This highlights the importance of these microorganisms to health and well-being. But by the same token, disturbance in the composition and physiology of these microorganisms has been linked to many human illnesses, from cancer to brain disorders to metabolic disorders, and we hope to prove that this may also be a case that HD may also have a altered gut microbiome, and that is exciting because that could give us an option, perhaps as a therapeutic avenue. So why is there an interest in gut microbiota in HD? We all probably know that Huntington disease is a genetically inherited disorder caused by expansion of this nucleotide in the DNA. It produces a protein with a long chain of amino acids that collapses on itself and aggregates. If you notice in this graph, the longer this repeats in the protein, the earlier the age of onset. But the majority of Huntington disease patients actually cluster in the repeats between around 40 to 50s. If you notice that in patients with even the same repeats, you notice that the age of onset is variable among these groups of patients. For example, some develop the disease in their 20s, others develop disease in the 60s. And this has been really perplexing and puzzling. And why is that? It's a genetically inherited disorder. You would have expected that the patient would more or less get the disease at the same age. So there are two possibilities. One is that genetic modifiers, that there are other genes in the humans that they could regulate the activity of this mutant Huntington protein for when to cause damage. For example, these patients here may have some protective genes, whereas these in here at the end of the spectrum here, that they may not have those genes, or they may have genes that they accelerate the disease. The other hypothesis is environment, and that is the concept of gene-environment interactions. Is the environment play a role in the progression of Huntington symptoms? So gut presents a very good environment. For one thing, it is really inflammatory, and it is a source of inflammation. And inflammation has been a environmental modifier in Huntington disease. Patients develop, inflammation in their brains many years before the onset of symptoms. So we are asking the question of whether changes in the gut microbiota may be a modifier of disease progression. So again, what are these gut microbes? And practically they control the entire human physiology. If you notice down here, every human more or less has about 100 trillion of diverse bacteria in the gut. This collection of the microbes again refers to as microbiota. It weighs about 3 pounds and is equivalent probably more or less to the weight of the brain. But it's very different than the brain because these collection of microbes have about 3 million genes. Humans have about 1% of these gene numbers. So in general, we are about 99% microbes and 1% human based on the gene dosage. What's interesting about this organ, which I call, is that the composition is adaptable, is subject to change. It changes with age, diet, medication, and also the host genetics, which is in the case of Huntington, which may affect the composition of these microbes. Some terms to remember as I go through this presentation. Dysbiosis is that when the homeostasis of these microbes change, when the composition or the physiology of these organisms change. And many factors are able to do that. For example, antibiotics, injury and infection, and other metabolic disorders do change this composition of these microbiota. Something to remember that this organ or this microbiota is here to serve us because, it informs important functions. And for example, they extract nutrients from the food we take in. They produce health-promoting molecules that are responsible for educating our immune system. In fact, you may already know that about 70% of our immune system gets educated in the gut environment by exposure to different components of this bacteria in the gut. And more importantly, they protect us against harmful bacteria. They prevent us from getting infection. And more important, again, that they control brain function. How do we acquire this complex gut microbiota? Actually, at birth, there are very, very few microbes present in the infants, and as the baby is born through the birth canal, it gets colonized by a specific class of microbes. And these bacteria are specialized in metabolizing the sugar molecules that they are present in the mother's milk. And as the infant grows and goes through dietary changes, they start acquiring different set of microbes. But all of these are really very highly orchestrated and very controlled. You don't just get randomly, specific any organism that you acquire. And it has to do with the selection of the host and the type of food practically we eat. So by the time of an adulthood, we have the most diverse composition of these microorganisms in the gut. And as we age, this diversity changes. There are some diseases that have been associated with this pattern of colonization. For example, early disturbances in colonization has been linked to autism spectrum disorders. And in the late age, dysfunction in the gut microbiome has been, linked to accelerated aging and some of the neurodegenerative disorders such as Alzheimer disease and Parkinson disease, which I will allude to later on. So how does gut bacteria may influence the brain's function? There are many, many possibilities and many pathways, and they are all listed here. One is that they produce neurotransmitters. These are regulators of brain activity. About 70 to 80% of serotonin, this is the happiness molecule, is actually produced in the gut environment. And again, these neurotransmitters are either produced by the gut microbes or they are stimulated by the gut microbes, so the human cells produce them. Also, the gut bacteria regulate the immune system. They could stimulate the immune system to produce either neuroprotective molecules or harmful bacteria. So if you have a balanced gut microbes, then it may produce neuroprotective agents, whereas if you have, in the case of an inflammatory disease environment in the gut, it may produce conditions that may lead to neuroinflammation. So bacteria also produce these small molecules called metabolites, and these metabolites produced in the gut could travel to the brain and promote expression of specific genes. And one specific one, a neurotrophin like BDNF, a brain-derived neurotrophic factor that is reduced in Huntington disease, is induced by bacteria and bacterial metabolite in the gut, and it's affected in the brain. So there are many pathways that these microbes could really communicate with the brain. And one important one is actually this vagus nerve. And if you've not heard it, this is the longest nerve in the body, is bidirectional. It receives signals, sends the brain to the gut, and as from the gut to the brain and from the brain to the gut, so it is bidirectional. So there are these direct pathways between the gut and brain that regulate this physiology. Not to forget that there is also an enteric nervous system. This enteric nervous system has about 500 million of neurons. It can function independently on its own, but at the same time, it's connected to the central nervous system and sends signals from the gut environment, whatever the bacteria send the signal to the brain. So there is, again, another pathway where gut and brain communicate. To note that Huntington is also, as also George mentioned, that it is expressed in the enteric nervous system. So a disturbance in the function of this enteric nervous system could also affect the central brain. So what regulates the diversity and physiology of gut microbes? Excuse me. As I mentioned earlier, drugs, antibiotics, and most importantly, diet can regulate the composition of these microbes. A lot of attention, and you probably have heard that a lot of attention has been paid to Mediterranean diet. Mediterranean diet is rich in components that seems to promote the diversity of the gut microbe. And this promotion of diversity has been linked to production of health-promoting molecules. These health-promoting molecules have been, again, associated with changes in gene expression in most organs, including the brain, and seem to have a positive effect on the human physiology. You got to remember that, again, diversity in the gut microbe is a signature of health and well-being. A lot of bad rep has been given to Western diet, and Western diet has been associated with less diverse microbiota, and a lot of inflammatory bacteria have been associated to be they've been shown to be propagated by this form of diet, and usually these microbes are, again, producing disease-causing molecules that changes also gene expression, but in a bad direction, that have been linked to many metabolic disorders, including cardiovascular disease and obesity. What's interesting here, that obesity is actually transmissible. That is, if you transfer microbiota from an obese person into a mouse that is germ-free, this mouse increases weight and becomes practically obese. In contrast, if you get microbes from a lean individual, and again, these studies were done with twins, and you transfer the microbiome of a lean person to the same mouse, and their identical diet, they remain lean. So there is something about the composition of these microbes, about the physiology of these microbes in the obese individual, that perpetuates that phenotype. Diet may also affect brain architecture and physiology. These are some studies that have been done recently with individuals on Mediterranean diet and those on Western diet. This red glow here, it demonstrates brain activity, practically measuring glucose. So if you notice that individuals on Mediterranean diet, they tend to have a higher brain activity compared to those on Western diet. This glow is much reduced. The arrow actually points to the area that have been affected in Alzheimer's disease. Gut microbiota could also affect brain architecture. There is a lot of shrinkage, at least in this case that they have studied in the brains of patients on Western diet. And in particular area that you see a lot of reduction in this white matter or the fibrils, which are also reduced in Huntington disease. So again, a word of caution here that these numbers are not, the N for these studies, they're not huge, but at least it points to a direction that perhaps sticking to a Mediterranean diet, the brain may benefit. And indeed, just yesterday, as part of the World Health Organization guidelines for reducing dementia, they highly recommend strict adherence to a Mediterranean diet, especially in the elderly and those at risk that may practically benefit from a Mediterranean diet. What happens with the diet, drugs, or other agents that cause changes in the homeostasis of this microbe? This changes obviously lead to what is called a leaky gut. I don't know if you heard this, but what happens in the leaky gut is the junction between the lining of the stomach. It expands, it makes practically an opening where the microbes could actually enter the circulation. Or microbial metabolite could enter the circulation and deposit in different parts of the body with the case of, stress or other disorders in the brain. Whereas in a healthy individual, you see this tight junction that, that these microbes and microbial metabolite cannot, penetrate and enter the circulation. So to be healthy, a very tight junction in the lining of the stomach is highly critical, and preserving this is a indicator of again, health and well-being. So again, leaky gut has been linked to a lot of neurodegenerative disorders. In the case of Alzheimer disease, I don't know if you're familiar, but Alzheimer patients, they get these deposits, these aggregated materials in their brain. They also have a lot of brain loss. So what happens in Alzheimer disease, they have noted that a lot of microbial proteins or other structures, microbial structures, that they are present in this amyloid plaques. So if you notice here, this red gunky stuff that you see here, these are microbial proteins lipoproteins known as lipopolysaccharide. This is actually a very potent bacterial inflammagen that causes inflammation in wherever it persists. So again, there is heavy deposition of this component in the brain of a subset of Alzheimer's disease patients, but other studies have shown that different set of bacteria and also viruses that they are present in the brains of Alzheimer's disease patients, and they have been linked to the progression of this amyloid deposit and brain damage. One interesting study that just came out a few months ago, that a microbe that actually causes gingivitis in the mouth has been detected in the brains of Alzheimer's disease patient. This specific microbe produces a toxin called a gingipin that again promotes actually the formation of these plaques and fibrillary tangles. And interestingly, these studies were able to make a drug that could stop this type of pathology in Alzheimer's disease models. Parkinson is also a very good example of gut dysbiosis, and you probably know that in Parkinson disease, a set of neurons in the midbrain are lost. Here is a healthy, normal individual. Here is one with Parkinson disease. You see this brown staining here in normal individual are lost in the Parkinson patient. So these are where the neuronal loss occur. And the neuronal loss is actually linked to formation of these protein aggregates, again, very similar something to Huntington disease. But in the case of Parkinson disease, they're called Lewy bodies. These Lewy bodies form in these neurons, and then the neurons die. There is a strong link between the gut inflammation and brain inflammation in Parkinson disease, and a lot of inflammatory bacteria and gut inflammation has been noted in Parkinson patients as well as in Parkinson models. There are studies undergoing that they actually think that Parkinson disease may actually initiate from the gut. So the protein that it gets aggregated in the brains of a Parkinson disease patient, they argue that could get aggregated in the gut and then migrate through different pathway to the brain, causing inflammation and neurodegeneration. These were some interesting studies that were actually done at Caltech in Dr. Sarkis Mazmanian's lab. What they did is they generated a germ-free mouse model of Parkinson disease. So these mice practically, live in a bubble without any germ. And what they found that these germ-free mouse models of Parkinson disease have very limited pathology compared to those that they have a set of microbes, like a normal mouse in their gut. So in other words, in this case, the presence of microbes in the gut of this mouse model is associated with the progression of Parkinson disease symptoms. What was very fascinating is that they got microbes from Parkinson disease patient, and they transplanted these microbes in a germ-free mouse model, and they saw an acceleration of the symptoms in terms of motor dysfunction and many other types of biomarkers that they look at, not stated here. But what was really interesting that, again, in these germ-free models, that the Lewy bodies or the aggregates, the protein aggregates that they form in Parkinson's disease, they were much reduced if they eliminated the microbe. Again, building this causation between the presence of specific microbes in the gut that may drive the symptoms of Parkinson's disease. What seems to be the underlying cause was the activation of these immune cells in the brain called microglia. In the context of the typical, microbiota, this microglia or, the immune cells in the brain were highly active compared to those in germ-free models. Other studies have shown that gut microbes actually control the formation of these brain immune cells. Indeed, germ-free mice have a different type of microglia compared to those that they have a set of, microbes in their gut. And the underlying component that regulates this gut-brain maturation of brain immune cells seems to be this metabolite that is produced by the microbes in the colon, and these metabolites are referred to as short-chain fatty acids and seem to be making a lot of news in terms of gut-brain interactions. So why am I saying this? That Huntington disease patients also seem to have elevated levels of these inflammatory cells in the brain. And this is actually in the pre-manifest stage. These yellow spots that you see in these brain scans, these are these activated microglia that seem to appear again in the pre-manifest stage in different parts of the brain as they have cut sections through the brain. This microglia are present. So again, we don't really know what's the connection between the microglia and HD pathology, but in a chronic disorder, like Huntington disease, it is likely that these microglia are hyperactivated, producing these inflammatory molecules that may lead to neuroinflammation. So is there a connection between gut inflammation and I have two inflammation there, and brain pathology in HD? I guess only time has to tell. We need time and research to explore that. Getting into gut bacteria in HD, is there gut dysbiosis in HD? And again, dysbiosis could be defined as a loss of beneficial microbes. And these are the good microbes that we need to survive or be healthy. Or there is an abundance of these pathogenic microbes that could be elevated, and again, leading to pathology in the brain. In our studies, we are attempting to use 3 different set of experiments. We're trying to look at the patient's microbiota. We're trying to look at HD mouse models, but more interestingly, we're starting to look at Drosophila model, which is very amenable to genetic manipulation to better understand the genetics of gut-brain interaction. To look at gut microbiota in HD patients, we are collaborating with Enroll HD in San Diego, Chile, under the auspices of Dr. Pedro Chana. So what we're doing here, we're attempting to collect fecal matters of various stages of Huntington disease, for example, pre-manifest, early HD, and late HD. We haven't got there yet, but we got some sample that I would share with you. But the way it's done, we collect fecal matters, we extract nucleic acids, and then we identify the type of microbes that they are present in this mixture of nucleic acid, because each microbe has a unique tag, so we could tell what type of microbes there are in them. So once a microbe is identified, obviously it doesn't mean much. We have to go and do translated in mouse models and to build the mechanism of how a change in the gut microbe may actually affect the progression of the symptoms. We have some early results that I would like to share you, and again, be alert that these are pilot studies. The N is only around 12. I'm only going to share this with you. What we have noticed that the diversity of gut microbes seems to be reduced in HD samples compared to the controls. Again, a warning sign, these are few numbers. We need a lot more numbers to make this assertion. But again, they are exciting. And again, as I alluded earlier, microbial diversity is associated with the gut health. So the seeing of the less diversity is really exciting to us. We are in the process of analyzing this data and including more samples from other sites that they are interested to collaborate, and we hope to get some more collaboration by giving this talk to make the story that whether there is a connection between the gut microbes and the HD pathology. But we are heading on with studies in the mouse models. In the mouse model is important because these are under controlled environment, under controlled diet, and there we would be able to make a better prediction of the gut-brain interactions. What we have seen in the mouse models, that we've seen that some bacteria are abundant in HD mice, but by the same token, there are some reduced. Some of them are actually in between. So it's very hard to make a prediction of what might be what at this point. What caught our attention that is that one class of bacteria that was altered in both mice and human. And that is really, I think, important because there may be a selection towards specific bacteria in the gut of HD patients. And again, it's too early to say, but we are hoping that ultimately this would lead to a answer. Going to the fly model, in the fly model, why do you even use the fly model? Flies are actually very interesting because they have a set of around 20 microbes in their gut. Most of them can be cultured and identified, so we know exactly what they are. And the important thing is that they are species that they are shared in fly and mammals, so we may be able to make prediction based on their physiology and composition. And I'm not trying to convince you here that Drosophila and mammalian intestine are the same, but there are some similarities in the anatomy. When we looked at the bacterial cultures between, a wild-type fly, and this is a fly that expresses wild-type Huntington and Huntington, if you notice, you see a lot of spots on this plate. This is a bacterial plate compared to the wild type. You see a lot more bacteria in the gut of HD flies. This is a very aggressive model, and the flies developed the disease real fast. What was interesting that the composition of these bacteria, that they were rich in inflammatory molecules. So some points to make here, that we've seen that potential dysbiosis in patients, in mouse and fly. And remember, in the fly, we're only expressing Huntington in the nervous system. That means the rest of the fly doesn't have Huntington. So we are thinking perhaps there may be a connection between the wild-type Huntington and the homeostasis of gut microbes. But again, more research is required. Again, seeing these results, we ask the question, what would happen if we eliminate the microbes in the gut of these HD flies? Again, this flies is an aggressive model. So we eliminated the microbes by treatment with antibiotic. We also give them some diets enriched. Practically, we gave them polyphenols that they are found in these types of foods, and they tend to have anti-inflammatory functions. Again, to remind you that HD patients also form these aggregates in the nucleus or elsewhere, that's well known. These aggregates could also be measured by different mechanisms. For example, in a gel, we could separate them and stain them with antibodies. These heavy black spots that you see, these are actually Huntington aggregates that they enter the gel and form this type of staining with antibodies. What's important to note here, that those flies treated with antibiotics have much less of this aggregated Huntington in their brains. And if we feed the flies polyphenols, a similar phenomenon happens, again, suggesting that the gut environment, at least in the fly, may regulate the aggregation of Huntington in the brain. What is more important, that manipulating the gut microbe also affected the mobility of these flies. If you notice this here, this is just allowing these flies to travel about 5 centimeters in a given time. Those that they were unaltered, we use as a control, they were less mobile. Only few of these flies were able to travel up here. Those that they were treated with antibiotics or polyphenol compounds, they were highly motile, and they were able to reach the top of this cylinder much faster than the control. Again, this results of obviously in the flies, but they make the point that perhaps gut-brain interaction may regulate the formation of these brain deposits, as well as the mobility of the HD flies. So seeing these results, we ask the reverse questions. What if we eliminate the gut microbes, again by antibiotics, but giving them the bacteria that we would like to do? For example, giving them inflammatory bacteria and see what happens to these flies. In this model, we use a less aggressive model, which the disease is progressive and occurs over time, but much slower than the previous model that I showed you. So the bacteria that we use is not important what they are, but they're present in the human gut. They are opportunistic, and they have Alzheimer disease and Parkinson disease. So we fed them these bacteria when we ask what happens to the inflammatory gene products. In a non-HD, and again expressing the wild-type Huntington, there was some induction of these inflammatory genes, But in the HD flies, it was about 4-fold higher. So in other words, it seems like the HD flies are hyperactive to inflammatory stimuli. And this is nothing uncommon. We've seen similar kind of, reactivity in the immune cells from HD patients. Then we ask what happens to the brains of these flies that they were fed this inflammatory bacteria. Again, if you compare the brain of a control fly to those that they were fed this inflammatory bacteria, you see these green spots, these green dots. These are the aggregated Huntington that we detected by antibody reactivity. And again, those flies that they were given this inflammatory bacteria had much more of these aggregated proteins, again indicating that the inflammatory bacteria in the gut may promote the aggregation of Huntington in the brain. Excuse me. So we also looked at their mobility in this model, and again those that is a less aggressive, so that we looked at them in a less earlier stage. Those that they don't have the inflammatory bacteria, they are more mobile compared to those that they were fed inflammatory bacteria, they are less mobile. So again, inflammatory bacteria, at least in the fly, seem to regulate the mobility as well as the formation of these aggregates. One more interesting aspect is that when we fed these inflammatory bacteria, we looked at the longevity of these flies, again, compared to the control. Those that they were fed the bacteria, they died much faster than the control. Again, the overall health and well-being of the HD flies, when given inflammatory bacteria, is worse when they don't have the inflammatory. So again, several aspects of HD pathology seems to be changed by gut microbiota in the HD flies. And these are, again, although in the fly model, they are very interesting in their own way, because mechanistically we could look into, say, what pathways between the gut and brain are altered that could influence this type of phenotype. What circuits are in there that could be influenced, and hopefully we could use those information to find drug targets that could be tested in other models. So again, do inflammatory gut bacteria affect HD? We don't really know at this point, but all we know that inflammation is present in the brains of HD patients. Again, gut inflammation is linked to brain inflammation. It's been linked to Parkinson disease. It's been linked to Alzheimer disease. And it would not really be surprising to me if, the same concept holds for Huntington disease patients. So again, in other models, for example, this is not a HD model. Would we be able to reduce brain inflammation by changing the gut? We have succeeded in other models that they have elevated this microglia, which are immune brain cells, and we treat them with a compound that drastically reduce the number of these dot green spots that you see in the brains of treated mice. So practically in the cartoon here, those that they have a lot of microglia, when we change their gut microbes, we're able to reduce the number of microglia in the brain. So the same concept holds true for other aspects of HD pathology, and I'm not showing the data here. For example, we could regulate the mobility, we could regulate the formation of these toxic proteins. And at the same token, by changing the gut environment, we could control the production of these molecules, such as the one I mentioned earlier, like brain-derived neurotrophic factors that has neuroprotective activity and is reduced in HD patients. So how could one go and approach, strategies to promote a healthy gut microbe in HDs? from an experimental point of view, there are several strategies underway. In the field of microbiota, a lot of scientists are trying to change the microbiota through nutrition. To me, this is the best approach because it's safe and is less risky. There are also the option of these medically tested probiotics. These probiotics are, again, live organisms that one could take and then change the gut microbiota. This is not to say that these are the ones that you find in your drugstore shelf. We are talking about medically tested and proven and FDA approved. And at the same time, regulating stress. Stress is highly linked to gut-brain interaction, and microbes actually sense stress hormones, and stress hormones change the composition of the gut microbes. So learning to regulate stress could be a physical way of practically changing the gut microbes. Giving you an example of how plant-derived molecules that could regulate gut microbe. Here is an example, ginger. For example, some elegant studies have shown that gingers are actually packed with these vesicles, that they are loaded with this nucleic acid. This nucleic acid, when ginger is digested, this vesicle packed with nucleic acid are ingested by selective gut bacteria in the colon, and this ingestion leads to production of metabolite that has protective effect. It has been shown to regulate immune system and the brain for the better function. It seemed to be linked to reduce microglial activation, and again, these are the immune cells, and it seems to inhibit the formation of amyloid plaque in Alzheimer disease. So ginger only represents, for example, one aspect of plant-derived modification of gut microbe. There are many, many others that could also serve the same function. One class of compounds that they gain a lot of attention in gut-brain studies are these polyphenols. These polyphenols seem to be enriched in this type of food, and they tend to have neuroprotective functions. They've been tested actually in various neurodegenerative disorder models, seem to have protective function. What these polyphenols seem to do is to activate the garbage disposal pathways in the brain, which is known as autophagy. And these pathways seem to participate in removing the aggregated proteins, which is common practically to all neurodegenerative disorders. So in terms of modification of gut microbiota as a therapy for HD, would that study lead to a magic bullet? We don't really know, and we have to wait and see if we could really treat or cure Huntington disease from the gut. But studies on gut microbiome in HD may lead to a better quality of life. And at some point, maybe we would be able to learn to delay the onset of disease by having a healthy microbiome. Again, imagine we're not here trying to cure through the gut, but perhaps even if we could bring the age of onset from 20s to 40s or 50s, that would be a major accomplishment to do. Something to remember again about gut microbiota modification, it's safety. It's very safe and really risk-free compared to a lot of chemicals that have been prescribed to other disorders. The expense is issue, and we would be able to cover a broader population of HD patients, and again, obviously, the ease of delivery. So in the past, we were told that, okay, we are what we eat, but we kind of really need to modify this statement. We have to say, actually, we are what we feed our gut microbes. And again, promoting a healthy gut microbe may benefit HD patients at any stage, and that could be easily, I wouldn't say easily done, but it's doable. So at the end, I would like to acknowledge the people that have participated in this studies, Angelica, Jung-Yun, Jan Ko, and our collaborator, Larry Marsh from UC Irvine. And I'm really grateful to HDSA for funding this research. We wouldn't have been able to do any of this research without their support. And as George mentioned, I've been fortunate to receive 2 times awards for these studies. And that's really, really a good thing. And I'm thankful. And thanks to all of you for listening. I'll be happy to answer any question that you may have.
Thanks, Ali. That was great.
Thank you, George.
Yeah, so folks, if you have any questions, feel free to type them into the chat function. Ali, I'll get started here. You mentioned in your, I know it's preliminary data, but you have a few samples from Chile that showed that the HD patients that were sampled had a less diverse microbiota. Can you define what that means? How are you determining what is a less diverse? Is it purely just the number of bacterium that you're able to observe in the sample, or is it something else?
That's exactly the point, George. Yeah, we look, the number of the bacteria, the overall composition of different phylum are different. So in other words, there are reduced bacteria and there are some that they are also missing. But it doesn't mean that there are not abundance of others. So we see some phylum, some classes that they are reduced in number, but others are also specific ones that they are, promote. So again, it's just based on the numbers at this point.
Great. You mentioned a lot. There's a lot of work that has been done in Alzheimer's and Parkinson's before the work that's now being done in HD. A lot of attention has been given to the gut and those diseases as opposed to HD. Are there any studies now that you're aware of that are, clinical studies that are trying to look at microbiome or gut microbiome modification as a potential therapy in those diseases that we could learn from for HD?
It's a very excellent question. The problem with, diseases like AD and PD is that, the disease has already started, and that's when we look into this. So we don't really know if any of those microbes that we find are causal or they're just a byproduct of the disease themselves. So in other words, if we do see some, let's say, elevation of this inflammatory bacteria, let's say in Parkinson, we don't know if these causing the disease or they are because of the disease that they are. So again, with the question of, chicken and egg, which one is coming first? But in animal models, as I was mentioned earlier, in animal models, if you transplant, let's say, the gut microbiome of PD patients into mouse models, you could accelerate the disease. So there is something in the gut that could promote symptoms. Yes, we could, look into those data and perhaps design some studies to see if there is a causal relationship between gut microbes and, the HD pathology. In fact, the studies that I presented on inflammatory bacteria in flies were based on those microbes that they are elevated in Parkinson disease patients. So, we could learn, but perhaps designing paradigms in models and see if that recapitulates some of the symptoms in the brain.
Thank you. Getting quite a few questions related to, obviously, different diets, whether it be vegan or, taking that versus the Mediterranean diet versus like specific probiotics that you could get over the counter at your local, CVS or Walgreens. There are a lot of questions. Would we recommend this for HD patients?
Again, the content of this presentation was not to suggest anything, and I would leave those up to participants themselves who participated in the studies. But it's common sense, to my opinion, to having a healthy gut microbes. Whatever that leads to a healthy gut microbe could definitely benefit the HD patients. Again, going back to what I would highly recommend that some of these participants, if they are interested, to read the World Health Organization recommendation for reducing dementia. Sticking to for example, Mediterranean diet has been linked to reducing dementia. Would that apply to HD patients? I don't know. But Mediterranean diet is an awesome diet that one could really enjoy. And if you enjoy Mediterranean diet, stick with it and, try to reap the benefits of it. But at this point, since we don't know much about the HD patients, we can't recommend really this diet or another. What I can say about the probiotics is that we don't know really anything about them. Those just been put on the shelf for the purpose of having, microbes, because even changes in the strain of microbes could have dramatic effect on their behavior, on their, physiology, and the effect on the, systems. So from what I have heard and from what I have learned by reading, most of these, probiotic, they have not been tested. But there were some studies, for example, at UCLA that they looked at, I think it was depression or some sort, I don't exactly remember, by just simply, feeding yogurt to the patients and looking at their brain activity. Those group of patients that they were fed yogurt, they had a better outcome than those that were not. So in terms of probiotics, if I had to take, I would love to take yogurts, and just because it tastes good and it's also beneficial. But I don't really know anything about the probiotics that they are on the shelf.
Right. Yeah, no, that's a good point. And a lot of these things that are on the shelf have not been, I don't know if they're not vetted and approved by the FDA. So not
at all. There are no, at this point, FDA approved probiotics that I know of that one could go into.
Yeah.
But again, a lot of natural products, a lot of fermentation products seem to have really, really good organisms that seem to be protective. I mean, a quote from actually Elie Metchnikoff, he predicted about 150 years ago that the future medicine would be by probiotics or eating yogurt. And I said, wow, he realized that 150 years ago. So maybe something about consuming yogurts.
Mm-hmm. Yeah, and it's a great point you brought up, and I got a note to myself to get the World Health Organization.
I actually have it on my computer. I will just send it to you.
That'd be great. Yeah, so I will get that and
just put it on, and then put it out there. And I think that would be an interesting, simply, written. So practically, you don't have to be a scientist to understand it.
Yeah. No, thank you. We'll take care of that after the call. I don't know. This is a good question, and I don't know if I have an answer for it. And Ali, I don't know if you'll have an answer for it, but it is because it's somewhat related because we're talking about gut, and this is less about gut microbiota, but there was a question from an attendee of today's webinar saying that they attended a convention, HSA convention, and thought heard that there was a correlation between overweight patients and a delay of the onset of symptoms. And has anything been learned subsequently about this? And I'm not sure if I have an answer for that. I know that doctors certainly, HD patients typically have a high metabolism and have a difficult time maintaining weight. So putting on the calories, packing on the pounds tends to be a good thing. But I don't know if there's anything more that's been learned about that. Do you have any insight in that, Ali?
I really have not heard. That's a very interesting point. I would love to get some information about that and who the presenter was. But I guess maybe there is a confusion between obesity and and weight gain. Maybe we need to be clarified, because what is known about obesity that highly linked to inflammation and brain dysfunction. I don't know if that's been. I don't even know if that's been done in HD patients, and maybe a rare case of here and there. I haven't heard of it, George.
Yeah.
But I would like to know who has presented and where the study's been done. That would be really cool.
Yeah. So we're running out of time, and there was one comment here, and I appreciate that comment, and we'll certainly do this, that when we do receive the WHO pamphlet, we forward a copy to today's participants. I am able to do that, so for everyone who did register and attended the meeting after the call, we should be able to send an email out with an attachment with this dietary recommendations from the World Health Organization. So we'll do that. Final question is, obviously there's a large component of, there's multi-components to your research in looking at the gut microbiota and biota and HD, looking in humans, mice, and flies. With the collecting human data, collecting human samples is obviously the most difficult thing for you. What can families do? Is there anything they can do now to help you?
Thank you for bringing this up, George. Absolutely. I would highly recommend to get, the centers that they are affiliated with, the HDSA Centers of Excellence, to kind of encourage the medical facility in there to participate in these type of studies, because, again, it may not be the ultimate, way to go for, curing Huntington disease, but it would ultimately make the life easier once we know more about the compositions of gut microbes in patients from different geographical locations. For example, these studies were only done in Chile. How do the gut microbes, look in HD populations in North America or in, East versus West? We're also, trying to get collection from other parts of the world. So what the participants could do, they actually encourage the centers to get involved and perhaps starting to collect samples and sending it to us. We would be more than happy to provide that information once we get the results. And again, participating in this research is the best help we can get.
Mm-hmm. And physicians, if families do have a physician or talking to a physician, and I'm certainly, of course, HDSA myself can help to facilitate this and having them connect with you, but should a physician have, this pique their interest, they should reach out directly to you?
Absolutely. Yeah, and my email is on the front page.
Fantastic. Mm-hmm. Well, excellent. Thank you, Ali, and thank you everyone for participating. Hope everyone's has a great rest of the day. Hopefully it's sunny and warm where you are. Our sun is finally out for one day in New York City. We're enjoying that. And like I said, we will not be having a webinar next month, but instead we'll all be up in Boston, June 29th for the research forum. So hopefully you can join us there, and if not, on the internet, where we'll be live streaming the entire morning. So thank you for participating. Thank you, Ali, for walking us through a really insightful slide deck and your work. I appreciate it, and we will be in touch, and we'll talk to you all next time we are on all together online. Thank you so much.
Thank you, George. Thank you all.
Bye-bye.